Students, and welcome to our today's revision, which is a biology paper 1 revision, West Kenya Union exam that we did uh, last term. That is the last exam that we had. So before we do that, allow me to pray. Heavenly Father, it's another wonderful moment we come before you. Even as we want to start this revision, may you help us, Father, to understand the concept and let the concept stick in our mind, even for future use. It is through the mighty name of Jesus Christ I do believe and pray. Amen. So once again, welcome everyone to our Biology Paper 1 revision session for Form 3. This is based on the West Kenya Union exams. It is good you test your understanding, and we shall do the questions one by one, revising the key topics and marking points for us to remember. Therefore, stay alert, take down notes, and ask questions where you are unsure. Okay, so we move to our first question. Let's move to our first question. Our first question is from the cell. The question says that give the structure of the cell that perform the following function. And our first structure is the structure that is responsible for the synthesis of ribosomes. Then part B, we were also to state the structure that helps in regulating exchange of substances in and out of the nucleus. And then part C, break down large molecules and destroy worn out organelles. Now, students, remember that every organelle has a specific job inside the cell. For example, the nucleolus, which makes ribosomes. Remember, ribosomes are the tiny factories that build proteins in the cell. The nuclear membrane is the one that controls the entering and leaving of substances in and out of the nucleus. Just the same way we have the cell membrane, which allows selective entrance and leaving of materials in and out of the cell. So the Golgi bodies then package and break down materials, kind of recycling center of the cell. So therefore, each part works together to keep the cell alive and efficient. Hence, the answers to our question, synthesis of ribosomes, we have nucleus, then the structure that helps in regulating exchange of substances in and out of the nucleus is nuclear membrane. And then the structure that helps in breaking down large molecules and destroy worn-out organelles is Golgi bodies. Next question. The next question comes from the topic ecology, which is our topic in Form 3. And the question states, give the meaning of the following terms as used in ecology. Give the meaning of the following terms as used in ecology. Now, in ecology, we study how living things interact with each other and their environment. Let's look at our first term, syncology. Syncology is basically the study of many species. Kind of studying the community. Remember, we have population and then we have community. Population is basically one type of species, but you see, community is a group of different species coming together to form a community. And if you study a community, that becomes syncology. What about carrying capacity? Carrying capacity is the maximum number of organisms an environment can support comfortably. Therefore, it's basically the maximum number of an organism an area can support comfortably without depletion of the available resources. Population. This is basically all members of a given species in a particular habitat. All members of a given species in a particular habitat. Therefore, ecology helps us understand the balance in nature and who depends on who. Next question. Question 3 comes from the topic nutrition. And the question is that the apparatus shown below was set up by Form 2 students in a certain school. So this is the apparatus. We can see this could be a boiling tube because this is some sort of uh, heat. Then we can see we have gas X being produced after some reaction here. Then we have hydrogen peroxide solution, which is coming to react with a less solution being added. So we have a gas being produced, and we can see the method of collection of the gas. Even how this gas is being collected can give you an idea of which gas it is, because we know this gas could be possibly be oxygen. Now, let's look at the first question. Name the gas. So we all know that when hydrogen peroxide solution reacts with catalase enzyme, that catalase breaks down the hydrogen peroxide into water and oxygen. Therefore, this gas is possibly oxygen. Hydrogen peroxide plus the enzyme catalase will give us water and oxygen. And let's come back to this part B. Write a word equation for the reaction. The question is word equation. Now, I was seeing most of the students were writing the chemical equation. There's a difference between chemical equation and word equation. When you are asked to give the word equation, please just use the words themselves. Hydrogen peroxide plus enzyme catalase will give us water and oxygen. Part C of the question. Where in the human body does this type of reaction occur? We all know where we conduct detoxification in the human body is basically where? In the liver. It's in the liver. In the liver. Okay. Let's move to our next question, which is a question coming from cell division. The question is study the diagram below and answer questions that follow. This is the diagram. We can see this is probably a cell and undergoing a type of division. We also have another cell here also undergoing a type of division. And we have something at the center of this cell too. And that thing is labeled as T. So as you are doing this paper or as you do any other paper, please always ensure that you note of the minor distinctions that take place or that appear in diagrammatic questions. Let's look at part 1. Identify the phase of cell division. You see, we have two nuclei form and the cell begins to split, forming a cell plate in plant cell. So basically, this one is telophase. Well, there was a kind of hint on T, but the question's answer was telophase. Part 2. Name the structure labeled T. Name the structure labeled T. I've told you, when we have this form of division, and you see at the middle of the cell, we have uh, a cell that is splitting and then forming something kind of a plate. This one is actually known as a cell plate. Cell plate. Therefore, the structure is known as cell plate. Identify with the reason the animal cell. Identify with the reason the animal cell. So cell 1, cell 1, cell 1. This is an animal cell. Cell 1 is the animal cell. Why? Because there's constriction of the cell membrane, which doesn't happen in cell 2, which is a plant cell. Remember, I've told you a cell plate will be formed in a plant cell. Then we also have presence of what? Centrioles. Presence of centrioles. presence of centrioles. So basically just understand that animal cells divide by constricting their membrane, while plant cells form a new what? A new wall. Okay, next question. Next question comes from circulatory system, and it is asking us to explain the significance of the following. Number one, why is the cardiac muscle myogenic? Actually, the cardiac muscle is myogenic so as to ensure that there is continuous and rhythmic beating without nervous stimulation. Now, in Form 4, when we shall be doing support and movement, you'll realize that we have some muscles which requires the permission of the brain to work or to perform their role. So, but we also have other muscles which do not require the permission of the brain to do so. These muscles are known as myogenic. And a good example of that muscle is the what? The cardiac muscle. Remember, cardiac muscles are the muscles found where? In the heart. Are the muscles found in the heart. Okay. 2. The left ventricle of a mammal heart is more muscular than the right ventricle. Okay, that is basically to generate high pressure to pump blood to the entire body. I usually say this, left ventricle pumps blood under higher pressure for longer distance. Higher pressure for longer distance as compared to the right ventricle. And you see, if you are doing that work, you must have some thick muscles, some thick and strong muscles. Most carbon dioxide is not dissolved in blood plasma. Why? So as to maintain what? The blood pH. It also prevents accumulation of acidity, and it is faster due to catalytic effect of carbonic anhydrase. You see, most carbon dioxide is not dissolved in plasma. It is carried as bicarbonate, which helps keep the blood's pH steady. Therefore, the system shows just how perfectly the body is designed to maintain life. Okay, let's look at our next question. The next question comes from the cell, and it's basically about centrioles. Let's look at part A. Give one function of centrioles. Uh, Actually, centrioles do form spindle fibers during cell division, as we've just seen from the previous question, that it is one of the characteristic feature in animal cell. Number 2, name a kingdom in which name a kingdom in which all members lack centrioles in their cell. You name a kingdom in which all members lack what? Centrioles in their cells. You see, centrioles is a characteristic feature of plant cell, but not of animal cell, but not plant cells. So if the members are supposed to have centrioles, then it basically means that these members belong to which kingdom? Animalia. Because all animals have animal cells and all animal cells have centrioles. All plants have plant cells. All plant cells lack centrioles. So members of kingdom Plantae, which are plant cells, do lack centrioles. Okay. Question 7 comes from circulatory system also, blood clotting. So study the flowchart below, which represent a physiological process in mammals. Okay, so we have prothrombin here. Then we have Y. Then we have X, which leads to thromboplastin. Then thromboplastin together with this Y, and it will change thrombin to thrombin, then thrombin to fibrinogen, and then fibrinogen to Z. So part A of our question, name the blood component represented by X. Which blood component is responsible for blood clotting? We all know that platelets is the blood component responsible for blood clotting. What is the significance of product represented by Z? Traps red blood cells, preventing further blood loss or entry of pathogens, or even heals the wound. And what is Z, by the way? Is known as what? Fibrin, fibrin, fibrin. Under what condition is thrombokinase released by the platelets? Under what condition is thrombokinase released by platelets? So thrombokinase is released by platelets only when a tissue is damaged and platelets are exposed to air. Next question from growth and development, that is metamorphosis. What is metamorphosis? This is basically change in body form during the life cycle of an organism. In most cases, we see insects changing from larva to pupa, pupa to adult. You see those stages, how the organism changes from one stage to another. That is what we call metamorphosis. Okay. What is the biological importance of the larval stage during metamorphosis? In larva stage, there is a vigorous feeding, hence the insects obtain enough what? Nutrients, or rather food. So it is biologically important because it reduces competition between larvae and adults. Each uses different food resources. Next question comes from reproduction. The diagram below represents one of the specialized cells found in the human body. Just by looking at the cell, we already know this is a what? A sperm cell, because sperm cell is characterized by the head, the neck region, and then the tail, So you see, this is the head. This one here is the head, the neck region, and then we have the tail. Identify the cell. The cell is sperm cell. B. Name the parts labeled B and L. Part B, which is the neck region, is known as the middle piece, And then part C is known as the tail piece, or rather the tail. What is the function of part labeled A? This is the part labeled A. Part labeled A has lytic enzyme which digests the wall of the ova. Actually, part A is usually known as what? Acrosome. Acrosome. The tail enables swimming towards the ova, a journey of survival and precision. Therefore, that is it. Let's look at question 10 about the teeth. So below is the dental formula of an organism. You can look at this. We have incisors 3, canines 1, premolars 4, and then molars 2, So we are to calculate the total number of teeth in the jaw of the animal. We have to calculate the total number of teeth in the jaw of the animal. So what you need to do in this calculation is that you have to understand that this formula is something that has been broken down to kind of a little or less digits, and it is usually division done by 2, divided by 2. So for you to get the total number of teeth, first of all you have to multiply the numerators by 2 and then also the denominators by 2. So basically this formula you multiply it by what? By 2. So the formula you multiply by 2. After that, you calculate the total number of teeth in the jaw by now taking the product of the numerator plus the product of the denominator, now getting the total. Now, when we work out, you see we have 3 times 2 Then we have 2 times 1, 4 times 1, 2 times 1. What do we get? We get uh, uh, we get 6 plus 2 plus 8 plus 4. This is the numerator, which totals to 20. Let's go to the denominator. You multiply 3 by 2, 2 by 1, 4 by 2, and then 3 by 2. What do you get? You get 6 plus 2 plus 8 plus 6, totaling to 22, which is the denominator. So we take the numerator, answer product of the numerator plus the product of the denominator, we get 42. Next question. With a reason, identify the type of dentition for the organism. Now, we have different types of dentition, but the most common one is heterodont, where we have a member having different types of teeth. For example, you and me, we are heterodont because we have, you see, incisors, canines, premolars, molars. So that is heterodont. Otherwise, we have homodonts, which are organisms that have similar or same type of question. Next comes from ecology, and it's talking of malaria. Name the organism that causes malaria. That is Plasmodium. Now, get it clearly. We have causal organism and the vector. The vector is the one that transmits, but the causal organism is the one that now gives the disease. So causal organism in malaria is Plasmodium, whereas the vector, which is the one that transmits malaria, is the female Anopheles mosquito. Check on the spelling. Female Anopheles mosquito. Not male, not any other type of mosquito, but rather female Anopheles mosquito. Control measures for malaria: draining stagnant water to destroy the breeding grounds, and then sleeping under treated mosquito nets, and of course using of mosquito repellents. Next question is talking about enzymes. Okay, the table below shows 3 enzymes A, B, and C and their respective optimum pH. So you look at the table, read the table. Name the most likely region of the alimentary canal of a mammal where enzyme B will be found. Check out enzyme B. What's the optimum pH? 2. Strong acid. Where do we have strong acid in the human body? Stomach. So probably that is the stomach. Then part 2, give a reason for your answer in one above. The reason is very simple. Enzyme B works best in acidic condition. Another reason is there's presence of hydrochloric acid in the stomach, which we all know that it is a strong acid that can amount to 2.0 as the pH reading. Next question is from gaseous exchange and specifically insects and fish. So we are to name the respiratory surface in insects. That is the tracheals. Remember, tracheal system in insect. Then we are also to name one feature that adapts the tracheal to perform its function. Number one, you see this, by the way, these structures, the respiratory surfaces are usually almost, the characteristics of respiratory surfaces are usually almost the same in all organisms. For example, look at numerous. Look at the respiratory surfaces in mammals, air sacs or alveolus. They are usually numerous, and that is to increase surface area for what? Gaseous exchange. Thin walls everywhere, even the skin as it is seen in frogs, it is usually thin for faster diffusion of gases. And then we have moist to dissolve gases. Why does the blood of insects lack hemoglobin? Insect blood is not involved to transport oxygen, and we know that is the specific function of hemoglobin that is usually packaging uh, substance that packages oxygen and transports oxygen in form of oxyhemoglobin. Oxyhemoglobin. Why do fish have gills that are highly vascularized? Why do fish have gills that are highly vascularized? Um, Fish have gills so that that are highly vascularized to increase and maintain a steep concentration gradient for efficient gaseous exchange. So fish have highly vascular structures so that they can maintain a steep diffusion gradient for oxygen uptake. Next question, which comes from classification 2. Classification 2. The diagram below represents a fan. The diagram below represents a fan. So we are to name parts labeled A, B, and C. So this is part A. Part A, you see, it is a magnification of this part, and then we have structures that have been labeled. And as I usually say, you look at the arrows. These are two lines pointing at two structures. So your answer should be in plural. Remember, if it is one, it is known as soras. But since there are two, they will be called sori. Sori. Okay. Part B is rhizome. As you can see, it is rhizome, something that gives the support. And then part C is the rhizoid, kind of roots, So that is it. And since it is a fan, of course, it belongs to division Pteridophyta. And you check the spelling, Pteridophyta. P is silent, but don't forget to put that P in your answer. All divisions, all kingdoms, all phylums, all other taxonomic units, their names begin with capital letter up to genus. It is only species name that is not capitalized, as we know from the rules of binomial nomenclature. Next question is about parasitism and specifically schistosoma. You are to state two ways in which schistosoma species is adapted to parasitic mode of life. Number one, it has two hosts, pigs and humans, for survival, lays many eggs to increase chances of survival also. It has thick elastic cuticle to protect it against digestive enzymes of the host. And then the eggs have a protective shell to survive harsh environmental conditions. And finally, their tissues are tolerant to low oxygen concentration, which is the characteristic of the gut. So you see, these features are the ones that will enable the parasite, which is schistosoma, to adapt in the environment where they are going to live. That is in the human alimentary canal. Okay, next question. 16. Transport in plants. The diagram below represents a transverse section of a plant organ. Study it and answer the question that follow. From which plant organ was the section obtained? Look at the structure. Look at the structure. Something like hairs, which are probably the root hairs. Look at the vascular bundle, how it is arranged. Something like a star shape, and this is basically the xylem. Then we have the phloems located on the arms of the xylem. This is basically a dicot root. Part B, give two reasons for your answer. Of course, we have presence of root hairs. This presence of root hairs will support our answer and our idea that this is a root. This is a root. But now the dicot part. The xylem is star-shaped. Phloem, the arms of the xylem. That is a characteristic now that will distinguish the monocot from the dicot root. Okay, question next. 17. It's about ecology also under population. So to estimate the population size of mosquitoes in the word is Nyansiongo village, visiting researchers caught 400 mosquitoes, which they marked and released. After 24 hours, 200 mosquitoes were caught, out of which 80 had the mark. So we are to do what? We are to estimate the population size of the mosquito in the village. So this is the formula. When we are calculating population, we take the number of the first marked organisms. So what is our first marked organism? 400. Multiply by the second capture, regardless of whether we have recaptured or not. So second capture, which is 200, then divide it by the captured and recaptured organisms. And that is why this method is known as capture-recapture method. So you see, from the 200 mosquitoes, 80 had been catched earlier on, marked and released to the same environment. So when you do that mathematics, we have a total of 1,000 mosquitoes. So remember, for you to count the 3 marks, we have first mark that is gotten here. First mark is actually gotten, and then we have the first mark gotten from the. Okay, so we have the first mark, and then we have those that have been recaptured. Let's look at our next question, ecology, and that is basically symbiosis. We are to name the bacteria found in the root nodules of leguminous plants. The only bacteria that is found in the root nodules of leguminous plants are usually known as the rhizobium bacteria. And remember, they are nitrogen fixing bacteria. So don't give your answer as nitrogen fixing bacteria. Even others go to an extent of just abbreviating as NFB, nitrogen fixing bacteria. No, that specific bacteria is known as rhizobium bacteria. What relationship is shown by the bacteria and the leguminous plant? That is basically a symbiotic type of relationship, or you can just say symbiosis. Next question. Explain how the following mechanisms hinder self-pollination. Protandry is basically a situation where the stamen matures early and shed their pollen grains before the stigma is ready to receive them. Therefore, you see in this scenario, we shall be hindering self-pollination. What about heterostyling? This is where there's different arrangement of the style and the stigma. So where we have the style is very different from where we have the stigma. So they cannot self-pollinate because they are differently arranged. So you see these both methods help promote cross-pollination, which leads to healthier offsprings. Next question. State 2 functions of muscles found in the alimentary canal of mammals. Number 1, they act as valves that close and open various parts of the canal. And then number 2, they control food movement in alimentary canal. You remember the wave-like movement, which is a characteristic of digestion in the alimentary canal. Next question. 21, a question again from nutrition and basically the liver. We are to mention where carbohydrates is found in abundance. So where is carbohydrates found in abundance in mammalian blood? Carbohydrates is found in abundance in the mammalian blood in form of what? Glucose. And then it is found in the liver in form of what? Glycogen. Now, you see, in blood, the carbohydrate is needed to be used, and it is needed in a readily available nature. that is glucose, readily available, the nutrient form. But you see, in mammalian liver is basically the storage form. How is it stored? And it is usually stored in the liver in form of glycogen. Remember the difference between carbohydrates in plants and animals. In animals, it is stored in form of glycogen, while in plants, it's stored in form of starch. Question 22. Liver damage leads to impaired digestion of fats. Explain. So liver produces bile, which contains bile salts that emulsify fats. The damaged liver won't produce bile, leading to the impaired digestion of what? Fats. So remember, we are justifying our answer. So in as much as we have already given the reason as to why, then justify your answer. Let's back to the question and tell us that due to this reason, it is what will lead to the impaired of digestion of fats. Okay? Next question. 23. Talking about blood groups. An individual belongs to blood group B plus or B positive. Name the antigen in the individual's blood group. We have antigen B and antigen D. Remember, that is something to do with the rhesus. So when we have rhesus positive, rhesus negative, so that is known as antigen D. Give the reason why the individual cannot receive blood from a blood group A donor. The reason is simple. A blood group A donor has the antibody B, which would correspond with the antigen B, leading to what? Agglutination. Agglutination. Okay, next question, 24. From ecology and basically eutrophication. It is the enrichment of water body by nutrients. So we will find that nowadays the water bodies have been deposited with so many decaying or decomposing matter, which increases the nutrients level of the nutrients level in the water. That is eutrophication. The effect of eutrophication in aquatic ecosystem, it leads to algal bloom. So we have too much of algae, and then we have depletion of oxygen due to increased decomposer activities. Next question, 5, which comes from gaseous exchange in mammals. We have this question stating that what is the significance of the following in gaseous exchange in mammals? Cartilage in the trachea. Cartilage, remember the rings of cartilage, which are usually in a circular manner, they do offer what? They do offer support and prevents the trachea from collapsing. What about moisture in the surface of alveoli? The moisture is there to dissolve the gases and allow them to diffuse in solution form. Like in the case of oxygen, it dissolves and then it is transported in form of solution. What about mucus in the inner surface of the trachea? Of course, the primary function of mucus in breathing is to trap dust and other solid particles. Trap dust and other solid particles. So all these features work together to make gas exchange efficient and protect the lungs from harm. Next question. Plant tissue, and the question is state 2 characteristics of parenchyma tissues. You see, the parenchyma tissues are usually good in buoyancy, and for them to perform that function of buoyancy, or for them to become buoyant, they must have thin cell wall first. You see, we are increasing the volume. If we are reducing the thickness, we are increasing the volume, and then we have large air spaces in the parenchyma tissues, and that's why I've said that they are kind of buoyant. And you see, most of the aquatic plants, and those plants that are found in swampy areas or those that are floating, they need to carry out gaseous exchange easily. So that only happens with the help of the parenchyma tissues. Aha, this is a perfect example of how plants adjust structure to function. In as much as these plants are found in the aquatic life or aquatic environment, they still carry out gaseous exchange easily. So class, this leads us to the end of our session, and I will urge you that you reach out for paper 2 so that we can revise again together. And with doing this, we shall ensure that we are grasping the content, the biological concepts, and ensure that we are preparing for our KCSE during that time when it comes. Keep safe. Ensure that you continue praying, that God continue giving you more wisdom and knowledge even as you prepare for KCSE. Otherwise, bye-bye. I want to wish you well. Say hi to your guardians and your parents back at home as we prepare to meet. Goodbye.